I have it on B. I put B for black. So if I go like that, it comes up. Yeah.
Wow.
You can do W too. It makes it go white.
Really?
Yeah.
That I didn't know that. And then what undoes it?
Just anytime you
Any click.
push the button or whatever you
Undoes
do.
B. Okay.
Undoes it, yeah.
That's great.
Yeah.
That's great. How you doing? Got Tom. Amy made it.
Yep.
Raj made it. It's good. This is a good turnout.
Yeah.
You got your goods?
Yeah.
All right.
Very kindly.
Cool.
Find my better.
Excellent. All right, we're ready.
Let it rip.
All right. Hi, everyone. Thank you for coming to another exciting Evos event. I am Glenn Garrett, the Director of Evolutionary Studies, and today I have the privilege of introducing our esteemed, renowned, and preeminent faculty member, Alex Bartholomew of our Geology department. Alex has been a member of the Evolutionary Studies Board since its inception, so he's literally on the ground floor of the program that we do like to consider the best academic program on campus. And he's been a very major force for the program, overseeing lots of evolution-related student collaborative research, digging up lots of fossils, and on one occasion he taught the Evolutionary Studies seminar course and gave it a nice geological spin. So Alex is an associate professor of geology. He has a very interesting academic background, a very prolific academic background. Last year, his book came out, Macroevolution in Deep Time. Doesn't that sound like a great book? Not a pamphlet, it's a book. It's at the library, in case you want to see it, or you can buy it, Amazon.com. And Alex is definitely a model of someone who I think makes learning fun. And that's something that comes across in lots of different things that he does. One of the classes he teaches is speleology, which I have come to learn means the scientific study of caves, which means he goes caving a lot. And on a few occasions, I was able to be part of a couple student groups where Alex brings people down into caves. For many students, it's the first time ever doing anything like that. And a lot of times people sort of scream and freak out, but no one gets hurt.
Nope.
And Alex can interpret every single facet of the cave. It's absolutely fascinating. He's got a PhD in geology from the University of Cincinnati. He's published extensively on the topic of Devonian fossils in particular. He also has some extracurricular interests I'll mention briefly. He is one of the best bagpipers in New York State. And is the director of Bagad, New York, the bagpipe band. And he has joined the Questionable Authorities on a few occasions, actually, playing the bagpipes. The first time I ever met him was at Bacchus, and it was a little faculty get-together after work. And I never met this guy before, and he says, "Oh, you're that guy interested in evolution?" I said, "Yeah," and he did one of these. He took right out of his pocket. He says, "Look at this rock. That's a trilobite from 400 million years ago. I found it on the street 2 minutes ago." I'm like, "Oh my God." And so that's kind of Alex. I'll give you one final anecdote before we bring him on. We did a great field trip a couple years ago up to Duck Pond with the Evolutionary Studies program, and Alex came along and interpreted things, and he's very good at saying, this is what this plant is, this is poison ivy versus grape leaves, look at these cool fossils. And then when we were up at the pond and we had a bunch of college students sitting there and kind of looking around at things, there were screams. And people look, and we see our friend Alex holding up about this big a dead corpse of a dripping, snapping turtle. And he pulled it out of the pond and was hanging it up, and people were freaking out. It was absolute mayhem.
I smelled for like a week.
Yeah, right. But
Wash that off.
But it definitely was worth it.
It was.
Yeah, so he's going to be talking to us about coordinated stasis today, and please join me in welcoming Alex Bartholomew.
Thank you. I'm excited to be here. Dr. Gare said I've been in on. Oh yeah. There we go. That's a little down here. As Dr. Gare said, I've been in on the Evos program since it started, but I have yet to give a talk in the Evos symposium, so I'm pretty excited to do that today. And today I'm going to be talking about evolutionary-ecological subunit turnover events. That's a fancy way of saying I look at fossils. And then amazingly, you can get a job doing that, which is marvelously fun. So as a bit of the outline for the talk, I'm going to set the problem. What am I trying to get at here? And then give a bit of background. Talk first about a little bit of macroevolution, species and above species level faunal change. And then a bit of geological background on New York State, where most of this work has been done. And then I'm going to talk about the patterns of faunal change that we see and some conclusions that I hopefully can draw from this. So first of all, why do I do what I do? This is why I like to do what I do. I've liked to look for fossils since I just about can remember. And it's a lot of fun, and New York State has an incredible fossil record. And I hate dinosaurs. I like to say that. I don't hate dinosaurs, but dinosaurs, they get all the women. So, trilobites are where it's at, man. Trilobites and brachiopods. Woo! All right? There's 5 or 6 of us who think this in the world. But these are all fossils that you can find in New York State. The beautiful trilobite, beautiful pyritized goniatite. Somebody who asked me about how fossils get preserved. This is one where the shell has been replaced by pyrite, fool's gold. Incredible shellfish and corals. Very diverse background of fossils in New York State. And looking at these fossils, these two dweebs here, Carl Brett and Gordon Baird, Carl was my advisor in graduate school, and Gordon might as well have been as well. He's at SUNY Fredonia, and Carl's out in Cincinnati. They worked tirelessly, unceasingly, documenting the fossils and the different layers of rock in New York State through several tens of millions of years. And they noticed a pattern. They noticed a pattern where the groups of fossils that are around, the groups of organisms, are there for a long period of time, and then all of a sudden something happened, and whole new groups of organisms were there. I said this before in the pre-talk, I'll say it again quickly here because it kind of makes a good analogy. All these families live on a street for several generations. Something happens, and 95% of the families move away, and they are replaced by a whole new group of families that live there for a long period of time. And then something else happens, and a whole new group of families move in. This is what they saw, except in these little things like Aleta fimbriata and Heliophyllum haughei and Phacops, all these different little things. This chart shows time on the y-axis and different fossils on the x-axis, and they're just showing where the fossils can be found in the different layers, and that they are the same species, unchanging really morphologically, and quite importantly, very few go extinct in this interval. Most everything that is around at the beginning is also around at the end, somewhere in the basin. So the question is, in the first 20 years of this was, the first 10 years really, this was put forth in the 1995 maybe, do we see this pattern in other places? Yes, we do. Okay. Now, what causes this pattern? What causes there to be stability in the intervals in the middle, and what causes these little changes at the boundaries? So my talk today is about those changes at the boundaries between these stable intervals. So first of all, a little bit about speciation, and I'm not going to lie, I scabbed this right out of my paleo lectures. So, here we go. Mutation within genes, within DNA lineages, causes variation amongst different organisms across the species. Sexual reproduction enhances this variation through the species. And then you have natural selection that acts upon that variation and acts upon the overpopulation of organisms to cause for the selective inheritance of fit characteristics. A very simplified version of kind of natural selection here. There are other factors that are going on as well, but let's just work with this. So if you look at how speciation occurs in the processes of speciation, you have evolution within a lineage of a new species. So you have a group of organisms that are alive, and somehow you develop a new species from or within that lineage. And you can have anagenesis, which is a pattern of gradual change of inherited characteristics. And really, we say that there's really not much in the way of diversity change. You're not evolving a new species along with an old one. It's just that an old one is sort of slowly changing into a new one. And then you have this idea of cladogenesis, where you have a lineage of organisms that are alive for a while, generation to generation, and then you split that lineage in two somehow. This increases the diversity. You now have a new species and the old species that is around. So anagenesis here, you have this kind of dopey picture of birds. All right, this one slowly inherits a number of changes through its genetic makeup that give it a longer tail and a red eye spot here. This one shows cladogenesis, where you have this species existing with not much in the way of morphological change, but at some point there's some divergence within that lineage, and now you have a new species. All right, and this change happens relatively quickly, and it's not like this where you're seeing morphological change over time in a direction, but it's sort of a quick jump, and then you have a new species. So, phyletic gradualism here is sort of like anagenesis. Most morphological change occurs after species splitting through this accumulation of change. And if you were to map this out through time, what you would see in the morphology is kind of this pattern of changing morphology over time after some kind of a species break. Punctuated equilibrium is really where most morphological change occurs with short bursts of cladogenesis here, and then we say little anagenetic change, so little morphological change over time. That is stasis, all right? So you have these changes that happen within the species lineages, and then they are relatively stable for a long period of time. Now, how can this happen? Well, a lot of it depends upon reproductive isolation. And how can we get reproductive isolation? You can have sympatric, is a fancy word of saying reproductive isolation within a larger population due to behavior. These two groups of animals, or the whole group, they can get in contact with one another, but a subgroup doesn't want to breed with another group. Or you can have geographic isolation of smaller reproducing species, or populations. This is called allopatric speciation. So just some diagrams to show this. I hate these diagrams, but they're from our geology book, and I hate them because time goes down. Time should always start at the bottom and go up. That's how it happens in the rocks, unless it's overturned. But here you have initial distribution of organisms, all right? Ecological types. I like to say fish on a reef. And you have those fish that come out at night and are around, swimming around. Hey, how you doing? All right, and then you have those fish that come out in the daytime. All right? And they never actually meet. So a night fish is highly unlikely to meet a day fish. So over time, all the night fish kind of breed together, and over time, the day fish breed together. Now, if they were to come out at the same time, they would be like, who are you? I've never seen you before. Oh, I've lived here my whole life. All right? So, but over time, you get these two separate populations. Because of the genetic drift and the mutations that happen are concentrated in those two smaller gene pools, even though they could technically come together. Allopatric speciation, you have two populations or maybe one big population with what we say gene flow. This guy here could go over and meet this lady here and produce offspring, all right? But something happens to create a geographic barrier between the two, and now a variant that pops up over here has a better chance of getting that, sort of variant morphology and variant genetics taking hold in this smaller population. And then, should the geographic barrier be removed, now the two cannot interbreed anymore. We can see this in the rock record through these wonderful illustrations I've prepared for you. Here's an island with shallow water and deep water, all right? And this brachipod, Mucrospirifer mucronatus, one of my all-time favorites. I like to call it the mustache brachipod. It looks like a mustache. All right? Sea level is high, and it's living all around the island. Something happens and sea level goes down. And now you have these little embayments where it's shallow, but it's too deep for the brachiopods to live all around this island. And the different Mucrospirifer populations, some of them get really long, some of them get kind of tall, and some of them get really squat, all right? You have genetic variation, differences in selection in these different little bays, and you develop different species. And now, when sea level goes back up, they can all mix and live in these different environments, but you have different species that are around. So, we also have to talk a little bit about mass extinctions. All right, mass extinctions are events that occur within the rock record where all of a sudden we see huge amount of die-off of lots of different evolutionary lineages. So we say that there are the big five through time here. This is time along the x-axis and number of genera along the y-axis here. This is 540 million years ago. This is 250 million years ago. This is 65 million years ago. This is now. All right, so it's not a linear scale along that bottom. It's just busted up into the different periods that we talk about. We have one here at the end of the Ordovician, one in the Devonian, a couple actually. The biggest one ever is not the one that killed the dinosaurs. That's the one over here. But this one at the end of the Permian where we lost 84% of our generic diversity died off, all right? Genus, Homo sapiens, all at the genus level, we had 84% die off of all these lineages. Went extinct, all right? For lots of different reasons through time, as it turns out. If we look at the Devonian in particular, this is that time interval that goes from about 417 to maybe 360 million years ago, an interval of time that I work on in particular. We have a number of extinction events. The big one here is this FF. If I back it up here, that's this one here. All right? That is this Kellwasser event. It's actually two events at the Fronian Fomenian, the FF boundary. But we have a number of them that go through the entire Devonian. And the middle and early upper Devonian are kind of chock with these things. It's a tough time to live in here. All right? Lots of extinction events happening. They tend to be tied to relatively high sea level in the Devonian, and they're worldwide in nature. We can find them all over the world. And what tends to happen when sea level comes up, the animals that are living in these different environments go extinct, and then new animals migrate in from somewhere else. All right? Nature abhors a vacuum, a wonderful statement I like to say. All right? A niche that is empty does not stay empty for very long. Something comes in to do that job. There's food there to be eaten. There's a life to be made. Something's going to move there and do that job. So we see extinction and replacement by new groups of organisms. So why am I giving this background? Well, it turns out that if I go to coordinated stasis and I'm looking at how these turnovers between these different groups of organisms occur, the patterns that I'm seeing are, to kind of jump the punchline here a little bit, a mixture of localized extinction and replacement and sort of, to use a loaded term, coordinated evolutionary turnovers within lineages. So I'll talk about that as we go through here. So the Devonian, a little bit of geologic background now. The interval that I'm looking at is the Silurian and the Devonian. Roughly 450 to 350 million years ago. About 100 million years, a turning point in Earth history, I like to say. One, because animals evolved jaws at this time. Before this time, you had some very simple invertebrates, or complex invertebrates that had simple types of jaws, but you didn't have vertebrate animals that had jaws, all right? Ways to eat things very, predatorially. Also on land, in the beginning of this time, we have very tiny vascular land plants that are maybe only 6 inches a foot tall, something like that. I hate the metric system, all right? And then you have, I love to say that I hate the metric system. I don't. We should all use a metric system, but whatever. I'll hold them to inches. Then, by the end of the Devonian, you have full, complete forest terrestrial ecosystems with trees that are 100 feet tall, all right? 30-meter tall trees. You have amphibians that are out on land that are 2 meters long. They're eating scorpions that are eating little bugs that are eating worms. So, complex land ecosystems evolve at this time. We don't have that before this. And we never see a complete wiping out of this again. So this is really a corner in Earth history. Before this time, not much, if anything, on land. After this time period, huge ecosystems on land. Now, the continents were different as well, and we talked a little bit about this in the pre-talk. Here's a reconstruction by a fellow named Ron Blakey, who is at University of Arizona. And these are, many of them are free on the internet for use. And he reconstructs where the continents were in the past. Plate tectonics, the continents move around. This area down here, this is not Pangaea, this is what we call Gondwana land. This is South America, Africa, Antarctica, India, Australia. All those continents are together as kind of one big supercontinent. Laurentia here is North America with parts of Europe. Here's Asia and Siberia, North China and South China. All right? And here's the equator. North America is turned sideways, almost 90 degrees, and it is south of the equator. So if we look, and here's New York State, right in here. So if we look at it as reconstructed in our way of north now being up, here's the equator, then, here's North America. We're sitting about 15, 20, 25 degrees south of the equator. We don't have any land masses west of really Nevada, all right? California, it's not there yet. Comes in later, gets attached on later. And areas sort of east of, say, Massachusetts are not there either. Florida has yet to attach itself to North America. Florida and Nova Scotia are actually parts of Africa, get left behind when we collide with Africa and then pull apart. It's a different story. But you have a big shallow sea that is here flooded into the continent. All right, this is the late part of the Silurian. Sea level goes up and down as well. That's another part of the story. Sea level can be low and the sea can be shallow, or sea level can be high and the sea can be very deep. Here's New York State. This is what we call a geological map. Shows all the counties outlined in their nice little dotted lines. Here's the wonderland that is Schoharie County, where I'm from. All right, here's Ulster County. We are sitting right about there. This yellow swath, that's the Gunks, okay? This green bit in here, this is all the Devonian, and the yellow is all of the Silurian. So almost 2/3 of New York State is covered by these Silurian and Devonian rocks. And there is an excellent exposure of all of these rocks. In other words, there's rock on the surface. I'm a horrible person to drive down the road with, because I never look at the road. I constantly, except when I'm driving the geology van, but I constantly look at the outcrops on the side of the road, and I'm talking and waving my head, and so there's lots and lots of outcrops around here. And the Finger Lakes are excellent. The Finger Lakes out in central New York State are cut down deeply, and there's hundreds of streams that go into them. Every stream has a huge couple hundred meters of shale and limestone that are exposed with excellent fossils. This is why this area has long been used as a testing ground for evolutionary ideas. We come up with a hypothesis of how we think things happened in the past and how life changed. One of the places to go to test it is here in New York State. So if we look at what the hell is this? This is time in New York, and all the different colored layers in here are where we have rock. And this is a great diagram, and it's hard to recognize what it is. Time is on the y-axis, and location in New York State is on the x-axis. Here's the Niagara region, here's the Finger Lakes, here's the Mohawk Valley and Catskill Mountains, here's kind of the Hudson Valley. Now, what are these peachy yellow areas in here? That is where there's no time recorded. The rock record is not perfect. There are missing chunks of time. Some people might recognize it. I like to make an analogy to the Watergate tapes, all right? There's missing time on the Watergate tapes. There's an unconformity in the sequence of events that happened, and that's what got Nixon into trouble, because he didn't have a complete succession of what happened. So that's what happens if we try and go and look at the Silurian in the Mohawk Valley. Most of it is missing. It's just not there. It was either not deposited or more likely eroded away. So what we actually see are rocks from about 405 million years ago sitting on rocks that are about 500 million years ago. So there's almost 100 million years of time missing. Now, if you go up to the outcrop, it's not like the rocks are here and then they're 100 feet higher. They're right smacked on top of one another. It's just that the time is missing between there. So that's where kind of doing the geology comes in to try and figure out where you have the best record. So what I'm looking at is this interval in here, in the Silurian and Devonian. You can see it's very well developed here in the east for the lower part of the Devonian, and then across New York State into the Finger Lakes region for the middle and upper Devonian. And the Silurian is well developed out there as well. And we actually have a pretty good record of parts of the Silurian in the east as well. So what did these environments look like? What did New York State look like back then? Well, this is the lower Devonian, and it shows a nice shallow sea. From tidal flat environments going out to kind of the zone of continuous wave activity at the beach, going offshore into deeper water environments where we say storm waves are the only thing that kind of touch down on the bottom. And lots of different organisms lived out here. Lots of little shells that we call brachiopods. They're not clams, but they look like clams. They have two valves, all right, but they feed a little bit differently. They're still around today. They're one of the most dominant groups and one of the best records that we have. You can have trilobites, which are arthropods, but they are now also an extinct group, but they were kind of creepy, crawly things. You had bryozoans and sponges and corals that lived in shallow water and were filter feeders, or we could argue micro-predators. And then gastropods, snails that crawled around, and some other weird groups as well. In the Middle Devonian, diversity increased. Diversity was very, very high. It's almost one of the times of highest marine diversity in the rock record overall as a whole. And that's because you had lots of different little environments. Remember that if you have one big environment, or one big ocean, and an animal can get to different parts of that ocean all at the same time, then you don't have the chance to get all this diversity. But if you can restrict the habitats into little patches around the globe, then you can increase diversity. So the Middle Devonian was kind of at the end of this time of more and more and more patchiness, and so species diversity was almost at an all-time high in the marine record. In other words, animals that lived in the ocean that were shells. This shows corals. This is a reconstruction of the seafloor from Chicago Museum. Big trilobites, cephalopods, all right, lots of different types of corals and neat things. Some little brachiopods sitting around here as well. So, what I'm going to focus on today is the lower Devonian and Middle Devonian and a little bit of the upper Devonian. So from about 400 and maybe 25 million years ago, I'm going to go down a little bit into the Silurian, to about 375 million years ago. So a span of about, I don't know, maybe 100 million years there, from the upper part of the Silurian into the upper Devonian. And if we look at the rock record, we divide it into a number of different layers. And those are called the Helderberg rocks, the Deer Park rocks, Sawkill, Southwood, Casanovian, here's a great word for it, Teafneoga out in central New York, and Taganic and Finger Lakes. And each of these has different layers of rock that are in there. And those different layers of rock have different types of animals. And the work of Carl Brett and Gordon Baird and other people have laid out a series of these stable blocks of fauna. The Helderberg fauna, which I'm now kind of, some of the work I'm showing, the work I'm doing is showing me that it can be kind of split into two. An Oriskany fauna, Asopus, Schoharie, Onondaga, Stony Hollow, named for the rocks just west of Kingston, Hamilton, Lower Tully, Upper Tully, and Geneseo. I'm going to show you what the fossils are in all these different groups in a minute or two. So the work that I've been doing is trying to look at what are the boundaries like between these blocks of time. People have examined the stability within them. One of the papers that I gave you to read was talking about the stability in this Hamilton interval and a statistical test to show that there really was strong stability of composition. Now, it's not abundance. And this was an argument that we went through for about 2 or 3 years back in the early 2000s in this idea of coordinated stasis, when we were trying to describe and figure out what exactly coordinated stasis meant. And Glenn asked me a great question. He said, is coordinated stasis a process or a pattern? And I think it really is a pattern. We're trying to get at what are the processes that control it, but it's really a descriptive pattern. And we've determined that it's a pattern at the compositional level, not abundance. So that means that if I go to the fossils and I go to the layers of rock, I'm not always going to see lots and lots of corals and a few brachiopods and like 1 or 2 trilobites. That's not going to be the way it is all the way through time. Sometimes I might find lots of brachiopods and just a few corals, and maybe a lot of trilobites and no brachiopods and a couple corals. That's abundance differences. The differences that we're seeing, or the similarities, are at who's there. What is the cast of characters? The fossils that show up at the beginning of the Hamilton time. I'll talk a little bit about this with some research I did with some students here at New Paltz, are the exact same animals, the exact same species, almost, well, we're dillying over time now. How long is this interval? But maybe 5 or 6 million years later, they are the exact same species. Right? The abundance shifts through time because you wouldn't expect all the same animals to live in all the same ways in every different type of environment because the environments change. But who's there is the same. So I wanted to attack this. What are the boundaries like between these different blocks of time? So I have determined that I see a type 1 boundary, I'm calling it, where you have concurrent lineage evolution. Groups of organisms that are around, and all the groups of organisms seem to, or nearly all, seem to undergo a speciation event within that lineage that is matched up at about the same time. Type 2 are my extinction incursion boundaries, and these tend to be concentrated in the Middle Devonian, where you have groups of organisms that are around, and then they all seem to go extinct, and a whole new group of organisms comes in and lives there for a while, and then they go extinct and are replaced again by a whole new group of organisms. So it's not that we're seeing, there are very few lineages that carry through across those boundaries. The things that are there, they just are gone from our area. And then, oddly enough, I have this one up here seems to be a mixture between the two boundaries. And, well, I'll save it for the end. Okay, so type 1, what do we see? Species A, B, C, D, and E are all happily chugging along through time. All right, time is on this axis here. And then they come to a boundary, and something happens, and we see their derivative lineages labeled as A prime, B prime, C prime, and so on. And they all seem to undergo speciation events. Now, some might go extinct, some might continue on, but we are able to tell that this species here derived from that species there. And it seems as though they all turn over at about the same time. Type 2, species A, B, C, D, and E, go extinct and are replaced by new species that come from outside of the area. 1 or 2, and I mean 1 or 2 over the course of 300, might continue on through, but most everything goes extinct and new stuff shows up. Obviously, the type 1-2 is a mixture of both of these. So, the lower Helderberg EE subunit boundaries, the lower Devonian, those between the Helderberg group down here, the Glen-Erie/Oriskany, the Esopus, Schoharie, and Onondaga. These all seem to be my type 1 type boundaries. Some taxa go extinct and some immigrate at each boundary, so some lineages just kaput and new things show up. But the dominant process of faunal change is concurrent within lineage evolution across multiple lineages. And here's the kicker here. This is often coeval with major sea level low stands. What does that mean? That's a fancy way of saying sea level got low. Sea level went down. We restrict the habitable area. And what I think I'm seeing is when sea level gets low and you restrict the habitable area, you have speciation events due to this maybe allopatric or sympatric, but predominantly probably allopatric speciation. Remember, that's the geographical isolate kind of population that occurs when you restrict the facies. And you do this across the entire basin, and multiple lineages go through sort of genetic bottlenecks. So if we look at the Helderberg fauna, we have some very nice brachiopods, some trilobites, all right? If you've ever been to Thatcher Park, New York, if you've ever driven along Route 209 in Kingston, just before you get to the mall, after you get off Route 28, you've driven past the Helderberg group. Very, very, very fossiliferous. Wonderful diversity of fossils in here. Several hundred different types of species. And they're living along for a long period of time. The Oriskany fauna, or Glen-Erie, named for Glen-Erie just north of Kingston on Route 32, where the Esopus Creek comes down along, well, 9W, along 9W there. You see fossils, organisms that have much thicker shells and are beefier in nature, all right? These fossils, these organisms were well adapted to live in kind of a shallow water, high energy environment. Then we go into the Esopus. If you've driven the thruway north to Catskill, you go past this thing that I like to call the abandoned thruway exit, a big shale deposit on the side of the road. There's some kind of weird radio tower there. You go in there and you can find some really rare little fossils and some really neat worm traces. But they're totally different than the fossils below. And they're different, again, from the fossils above in the Schoharie formation. Now, they are related groups in both of these, like this one here comes from this lineage, but they're totally different. So, looking at this, I've color-coded these for the time periods: Helderberg, lower, Helderberg, upper, Oriskany, Esopus, Schoharie, Onondaga. This kind of rainbow pattern goes through the whole thing. You can see you have the brachipod species Howellella vanuxami that goes into acrosphere for Cycloptera. That one is derived from that one. Acrosphere for Murchisoni derived from Cycloptera, so on and so forth. If you look at another group of brachiopods, all right, the Rensselaria lineage, you have Rensselaria that comes from Nanothyris down here, that goes into Edymothyris through here, that goes into Amphigenia small here, and then a large Amphigenia up here. You see that these speciation events between these brachiopods, I took this from guys who know brachiopods, all right? So these are people who've done this work and demonstrated that these brachiopods are all related to one another. And they said, here's where I first find them, here's where I last find them, that, so on and so forth. So I'm synthesizing all this data together and recognizing and pulling out this pattern of coeval turnover events. These were the first two groups that really kind of tipped me off to this. And then I went into the rock record and looked more. And I find that in Oklahoma, in the same period of time, the natives are restless. People don't like this. I'm going to keep going. All right, in Oklahoma, you see the same pattern through the same periods of time. We can correlate between these two areas, and we can say that these rocks are the same age as these rocks over here, and that they, again, see, here's Dalgina, Henryhausensis, Oblata, Musculosa. These brachiopods, the species turnovers are at the same time. Here you have a group of orthid brachiopods, and this actually Reeftonia, this boundary, this is in New Zealand, not even in New York State, all right? So you see that the boundary between these things match up to these time periods, the same turnovers that we have in New York. Here are mutationellid brachiopods from up in Maine and New Brunswick and Quebec. And again, you see the patterns of turnover match up generally pretty nicely closely. Not always, all right, and some of them sort of go extinct in different places, but the patterns are matching up here. Centronellids, brachiopods. Again, same pattern, all right? I'm not going to bore you with any more of these. Well, one more, maybe 2 or 3. Atropaceans, canidids, all right? All of these things are showing the same pattern of coeval. I don't like to use the word coordinated, it's a loaded term, but coeval turnover at these boundaries. Middle Devonian, all right? We get to the Middle Devonian. Different type of pattern. This is an outcrop in Leeds, just north of Catskill, or just west and a little bit south of Catskill, actually, along Catterskill Creek. This is Chuck Verstraeten. He's a New Paltz alum who now is one of the state geologists at the New York State Museum, and my friend Mike DeSantis. And they are sitting at the contact between the Onondaga limestone and the Marcellus shale. All right, we all have heard about that. Full of just evil gas and oil. So, but this contact is a major faunal turnover event. All right? The fossils that are down here are very different than the fossils that are up here. Type 2 boundaries dominate in the Middle Devonian between the Onondaga, Stony Hollow, Hamilton, Lower Tully, and Upper Tully. And this data here, these CAHO, what does that mean? Carryover and holdover. That means that of all of the 209 species in the Onondaga, only 2.4% of those make it into the Stony Hollow. And the Stony Hollow fauna is comprised of 11.4% of the taxa from the Onondaga. That's what holdover means. It's a very simple statistical thing, just how much of this comes from here and how much of this goes on into there. Stony Hollow into Hamilton, only 2.6% make it into the Hamilton time. All right? So on and so forth. So you can see these are very distinct faunal boundaries. And in each case, we're seeing an incursion of what we call old world realm faunas. Things go extinct in our basin, and stuff shows up from somewhere else. Oh, and these are coeval with major sea level flooding surfaces. So as opposed to sea level going down, sea level's gone up now. So, Onondaga into the Stony Hollow. Beautiful fossils in the Onondaga Formation. Who's been to Five Guys in Kingston? A couple people. McDonald's? All right, well, next time you get your burger, go out back and look at the Onondaga. All right? It's the first stop on our second geology field trip in my Strat Sed course, is the McDonald's in Kingston. I go get my McGriddle, which is the tastiest sandwich ever made. And then, I have yet to try that taco thing. I really want it. But then we go out on the outcrop and we find corals and brachiopods and we just have a grand old time. All right? And then from there we drive around the corner and we go to the side of Route 209 in Kingston. And Katie and I spent a whole week on this outcrop and dangled off of ropes and nearly killed ourselves and stuff. But we were trying to find as many different types of fossils in this as we possibly could. And we didn't find much, but almost everything that we found was totally different than what was here in the Onondaga Formation. So what's happening then? This is a case of one of these, sea level goes up, and it gets so deep, the water gets so high, that the reason we have black shale in the Marcellus shale is because sea level gets so high that there's no oxygen in the bottom water, and all of the things that are alive in the water column sink down to the bottom. And instead of getting eaten up by bacteria, somebody asked about fossilization. Instead of getting eaten up by bacteria, the organic matter gets preserved, and the weight percent, up to 20% of the weight of a piece of shale is organic matter in this Marcellus shale. So when the oxygen is so low in the water, these animals that live on the seafloor, they just can't live. They cannot survive in this area. They are somewhere. They have to be somewhere, because it's not a worldwide extinction, but locally, in the Michigan Basin, in the Illinois Basin, in the Appalachian Basin, it is an extinction event. And then what happens when sea level goes back down a little bit? Well, we say, connected our basin up here to, and this is one of my favorite things in geology, the equatorial Arctic. All right, in the Devonian, the Arctic was up at the equator, so we have these warm water faunas that existed in what is now the Arctic at the equator, and they come to New York. All right, we can look at the fossils that are up here in older times, before the extinction occurred here, and after the extinction, they make their way into the Michigan Basin, into Iowa, into Illinois, and into New York. And actually some stuff does an end run around here as well. And then what happens? Well, then things change back into the Hamilton group. Sea level goes back down, and actually what we see is a bunch of stuff comes out of South America and makes its way up into our area. Sea level goes back down, it gets colder, and these warm water adapted taxa that were living in the Canadian equatorial Arctic that moved into our area go extinct and are replaced by cold water fauna that come in like this. One brachipod in particular is Tropidoleptis, which is not, this one right here. And a famous German, or I think he was Spanish paleontologist said, yeah, this is the Nazi brachipod. And I was like, what? All right, and he said, yeah, well, it goes from Germany to Canada to South America and then to America. And then back to Germany at the end. And I was like, all right, dude, you're crazy. So, but I never forgot the route that Tropidoleptis took through time. All right, so Tropidoleptis is a cooler water adapted taxa that makes its way into North America, indicating that sea level has gone back down and it's gotten a little bit colder. And that fauna of the Canadian equatorial Arctic, totally gone. Totally gone. Very good exposures of this in the Finger Lakes region and out on the Lake Erie shoreline. And then you go into the Tully Formation, Taganic Falls, beautiful waterfalls, west side of Cayuga Lake, just northwest of Ithaca. At the bottom of it is this nice limestone, and that has a different fauna. What is it again? Sea level got high at the end of the Hamilton group, and another warm water fauna emigrated. Then, out of the Tully into what's called the Ithaca, or Genesee fauna, I see a mixture. I see both some things that go extinct, and I see some evolutionary lineage changes. This is a bit of a weird one. And this has to do with the fact that when sea level went up, we also saw a progradation, a building out of the Catskill Delta at this time. This is a little bit complicated, but just needless to say, not all of the environments were flooded out, but the environments became restricted when sea level went up. And those animals that lived in those restricted environments underwent a kind of coordinated coeval speciation events, and out in the basin we had replacement by new things that came in. So this is most likely what's happening all the time, I would argue. Somewhere in the basin we're getting a type 2, somewhere in the basin we're getting a type 1, all right? But this one is actually a place where we can see the mixture happening between both of them. So if we match these up to our sea level curve, and here are our faunas, Helderberg, Oriskany, Asopus, Schoharie, Onondaga, white is Stony Hollow, purple is Hamilton, blue Lower Tully, and then kind of brown is the Upper Tully into the Geneseo. We see that these here, generally these boundaries are related to sea level low stands. In the Middle Devonian, they're related to flooding events, and in the Upper Tully, it's kind of a mixture of both. So, the EE subunit boundaries, these boundaries between these stable blocks, different processes of faunal change are occurring. Type 1 is associated with sea level low stand and possible restriction of habitable area and sort of allopatric speciation events with geographic restriction and genetic bottlenecks across multiple groups of taxa. Boundary 2, type 2, in the Middle Devonian, is extinction incursion. Sea level goes up, just kills off everything that's there, and when sea level comes back down a little bit, new things move in from outside of the basin. And then the type 1 and 2 boundary is a mixture of the two. In some parts of the basin, everything goes extinct, new stuff moves in. In other parts of the basin, where the restriction of facies occurred, we have concurrent evolution. So, that's it. And I'm just done in time. Happy to take questions. Raj. Probably ought to. The first life on land, it was probably algae, or quote unquote algae, thin little layers of algae. The oldest, we have evidence for, we don't have body fossils, so we don't have pieces of grass or something. What we actually see are the spores, like pollen-like things, indicating that there was life on land in plant form. And then the first animal that came out on land, if anybody's in my historical geology class, is a question on the test, is a millipede-like fossil that we find in Pennsylvania in the Ordovician. Yeah. Either I totally baffled you or you know everything. Yes?
Can you say something more about the boundaries of level rise?
Mm-hmm.
Does that mean the land?
So what's causing the sea level change? There are a number of different things that can drive sea level change. Some of it can be climatically induced. If I back up just one slide here, wrong way, to this one. Between the Stony Hollow and Hamilton time, we see a pretty big sea level rise. And that is kind of driven by multiple things. One is that this is a time of mountain building on our east coast. And so the floor of the basin actually sinks down. It's like walking out to the end of a diving board, and the diving board goes down a little bit. So you actually push the land down a little bit. We also know, by looking at some complicated ways of temperature data, by looking at teeth of certain organisms and looking at the isotopes, we can tell that it actually got warmer at that time as well. So it's probably this double effect of the land went down, and it got warmer, and any glaciers that were around melted, and sea level went up. When sea level goes down, either maybe it got colder and water is getting taken out of the oceans and tied up on land as ice, or it's possible that you can, another kind of large-scale driver of sea level change is the rate at which seafloor spread. If you have very rapid rates of ocean floor spreading, sea level is going to be high because the crust that is produced is warm, and it's kind of warm farther from the ridge, and so it makes the overall the oceans are shallower. But if seafloor spreading is slow, then the crust is kind of cold and it sinks further down and you get deeper oceans. And that's actually going to make sea level sort of go down. It's a little bit of a complicated process. Remember that we are looking at seas that are on land. They're on the continent. So while the ocean basin might get deeper, that's just going to pull the water off land, and all the water is going to go into the ocean. We're actually looking at what we call, fancy word here, epicontinental sea. It's like the Black Sea. It's where you have the water that's pushed up onto land, so to speak. Yes? Oh, yes. Yeah. The oldest fossil forests in the world are from right about here. All right? You have a plant record that starts to get pretty good right around here. And the oldest big forests are from southernmost Schoharie County in Gilboa, New York. And not only do you have fossil tree stumps that are big and trees that were maybe 30 feet tall, but you have the oldest spiders, the oldest centipedes, the oldest gnats, some of the oldest insects. You have scorpions. You have a bunch of worms and lots of different types of plants. So one question is, with the change that's occurring on land, is any of that affecting what we see happening in the oceans? And that's a super good question, but it's going to take a lot of work to figure that out. But that's kind of a long-range goal of mine, something I've always wondered about, because I did my master's thesis on these Gilboa forests and looking at how they were preserved, and it really got me thinking, what's the tie between land and the ocean? There has to be some tie there. But we don't have a good enough resolution to say, this layer of rock on land equals this layer of rock in the ocean. That's where our breakdown is at the moment. So we're trying to work that out. Yes?
Even without knowing the exact as well as
Yeah. This is one thing I noticed by looking at a paper in grad school. As I came in one day and I was pawing through, and there are a number of floras, these intervals of time where you have similar land plants. And at a very rough, like I'm going to tape it on the wall over there and stand here and look at it and squint my eyes, it looks like they match up. Needs a lot of work, but I think so. Yes? No. Okay. So she asks, how at these boundaries do the sessile things, like corals, things that don't move around, how do they survive? The sea level change isn't drastic. It's over the course of days and years and months and millions of years. So from generation to generation, they send up their little larvae into the water column, and they float around, and they come down where they want to live, and then they live there. Now, if you restrict it too much, or if you just don't have that facies, that nice shallow water, clean stuff preserved, they're just not going to be there. Now, in this Hamilton time, one thing that we know is that there are beds that have corals all across New York State. And then there's like 100 feet of shale where there's no corals anywhere. Where do those corals go? That's a good question. And we always invoke, well, they went to Canada. All right? Somewhere up there had to have been an environment that was habitable for them. And then when their environment came back in New York State, they said, ooh, let's go to New York for the millennia. All right? So, yeah, these, as I said in the earlier talk, these taxa, these animals, they track their preferred environment. They're going to like to live where they like to live. All right? And so if it gets deep, they're going to move and follow the beach around. Or if it gets shallow and they like to live in the deep water, they're going to move back to where the deep water is. Now, if all of the shallow water is gone, that's when there's an extinction in the basin. If the sea level gets so high that the entire basin is flooded out, then there's nowhere for them to go, and then they die off. Right, good question. So, this is the big end Devonian mass extinction. I have to say that I disagree with you.
Oh, I wasn't asking.
No, I know, but I disagree with what you're asking. Because you're asking, isn't that where the end Devonian, or the Devonian mass extinction starts to happen? No. The Gilboa forest is here in the Middle Varkas zone. The end Devonian mass extinction, and that's maybe, I have a big problem with these timescales personally. The Emsian, this is like such a boring period of time, and it's 17, give me a break, it's not 17 million years long, it can't be. All right, but that's another soapbox. So the end Devonian mass extinction is up here, it's 20 million years later. There are a number of extinction events that occur, but each one is its own individualized event. Now, if you were to say, is that the time of a middle to late Devonian biocrisis, I might say, maybe, but I don't know if we can tie right now between the two. A hypothesis has been thrown out by a guy named Tom Algeo, who I had as a professor in grad school, that said, well, these forests, as you said, acidification, that you change from having no chemical weathering to having intense chemical weathering on land. The chemical components that are getting into the ocean water are totally different at this time. That's what's causing the big mass extinction and all the black shales. Maybe over time that's possible. But, and you asked me this earlier today, and it tweaked me a little bit. But, anyway, I think that the Kelvasser events are their own thing. The Gilboa forest is down here around the Pumilio events. So it's just a little bit different. Yeah. There was no map. There was no map. This one. Oh, okay. Where is Tiktaalik coming from? Yes, yeah. Irish. No, Tiktaalik is found in terrestrial deposits. Yeah. Yeah. Tiktaalik is younger than our rocks here. Now, we have a big pile of terrestrial rocks in New York State. Once you get up in the Catskills above, if you're driving from Kingston towards Phoenicia, once you get past West Hurley, it's all non-marine. It's all terrestrial. And those deposits are there, but they're just a little bit too old to have vertebrate that are on land. Some of the oldest ones come from a place called Red Hill in Pennsylvania. You can Hynurpaton means Hyner crawler. Hyner is the town of Pennsylvania there. So in that is, I think that is latest Fronian, somewhere up in here. So that's some of the oldest terrestrial vertebrates that we know. Yeah. We have a lot of fish in the Catskills. There are a lot of good fossil fish, and there are lungfish, and that have estivation burrows, and there are some lobefin fish as well, but no terrestrial vertebrates. Yeah. Yes? Mm-hmm. Good question. People have argued that we are in the 6th mass extinction, the Anthropocenic mass extinction or something. Probably because we as a species are carrying other things all around the world. Snakes get on a plane, and people get really, really upset. And then that plane lands at an island, and those snakes get off the plane, and then those snakes go eat those birds. All right, so that we are causing mass extinctions. I should show that movie in class and be like, extinction. All right, but yeah, if sea level were to come up, New York City would be underwater. Albany is at sea level. Tide comes all the way up to Albany. Parts of Albany would be underwater. We'd lose most of Florida if sea level came up, I think, 5 meters. Most all of Florida would be gone. Sea level has gone down a lot recently, or in the recent past. A friend of mine used to be a bay clammer in New Jersey, and now he works on fossil sharks. And he has buddies who do dredging for clams 40 miles offshore of New Jersey. And they're dredging, and they pull up a cervical vertebrae, a neck vertebrae, of an elk moose 40 miles offshore. That means that maybe 12,000 years ago, sea level was down enough so that 40 miles offshore was exposed land, and there were a giant moose walking around. All right? So, yeah, if the glaciers were to melt more, if the West Antarctic ice sheet were to calve off and all melt, sea level could come up. I don't know exactly how fast the rate of rise is. That's a tricky thing to predict. But we do know that it has happened in the past because we see these faunal events that indicate that you had water that connected from New York all the way up to Alaska. But then part of the time, there was a distinct barrier between those two land masses. So it's hard to predict, but it's a good question. And sure enough, sea level will go up and sea level will go down. That'll continue to happen in the future. Any other questions? Time for the barbecue. Thank you.